All right, hi everyone. We'll give it a few seconds here while folks trickle in. We are excited for our first AYAO power session. And before we get to the fun part of why you all are here, I'm going to go over just some technology comments and questions for folks real quick. And for those that don't know me, I'm Nick Galrakis, the Executive Director and co-founder of Adolescent. So welcome to our first session today. We're honored to be with some amazing individuals on this new partnership. I'm going to make my part short and sweet and then pass it over to Mike Roth to give the introduction of the AYAO Power as well as today's session. So as a reminder, for those that have been on Zoom before, you know the drill, but for those that might not be as familiar with Zoom, we do have a Q&A area. We ask that you ask questions for our presenter or any questions at all in that Q&A space. This way, it's easy for us and our moderators to be able to get in there, ask those questions at the end. Chat is open, so you can go ahead and just have a conversation in chat if you choose to do so. But again, it's just easier in the Q&A area for us to keep track of questions and not have to worry about if it's in the chat space. So if you put a question in the chat space, we might lose it. So definitely make sure it's in the Q&A area. Secondly, we've gotten a lot of emails from folks that have registered. If this was going to be recorded, absolutely it's going to be recorded. This will be available to folks. We will notify all registrants once it is available. So this way, you know to go back to grab that, as well as resources that will go with this video as well. That's going to be on that main hub space on elephantsandtea.com's website. So we'll have the link. We'll make it very simple for folks to get into it. Lisa just put the link on there. That will be our home base for all recordings and resources that are part of this amazing series. So with that, I just wanted to get those few things out of the way. We are currently recording now, so everyone's aware of that. And I'm going to pass it over to my friend Mike Roth to take it from here.
Great, thanks so much, Nick. And really just excited to welcome all of you guys to AYAO Power. So to give you a very brief background before we get to our first talk of the curriculum, AYAO Power is a partnership between the AYA Oncology Education Working Group and Adolescent. And really what we're trying to do is fill a large gap in education and knowledge on the needs of adolescents and young adults diagnosed and treated for cancer. We did a study last year looking at education for the AYA oncology population during oncology training. And what we found is that few oncologists, pediatric medical oncologists, as well as other trainees ever receive any education on relevant topics for this younger population. So we decided, why not now fill that gap with a remote curriculum that's novel, that is easy to access, and brings expert speakers really right to your computer, right to your phone. This is going to be an annual curriculum. The speakers are going to change every single month. The content is going to live centrally. For your colleagues, if they can't make the talk live, please have them go to the central hub afterwards. The goal really is to make sure that all of our providers feel comfortable taking care of AYAs during their career while patients are on treatment or off treatment. So with that very brief introduction, I want to thank a few people. One, thanks to Nick, Kayla, and the Adolescent group. Thanks to my partners, Scott Merdler, Dr. Hayes-Lattin as well. They are key members of the AYA Oncology Education Working Group and have helped organize all of this. And yeah, let's start the curriculum. So it's a real privilege to introduce our first speaker, Dr. Brandon Hayes-Lattin. Many in the AYA world know Brandon quite well. He has been an advocate and really a driver of the AYA mission for I won't age him, but maybe a couple decades now. So currently he serves as a professor of medicine and deputy division head, Division of Hematology and Medical Oncology at Oregon Health and Science University. He's also the director of the OHSU Knight Cancer Institute's AYA Oncology Program. He really is an expert in all things AYA oncology, and I'm just thrilled to pass it over to Brandon to kick off our educational curriculum. So Brandon, it's all yours.
Mike, Nick, everyone, thank you very much. Thanks for joining today. I'm going to pull up my slides real fast. Okay. So welcome everyone to the kickoff talk for AYA Power. I am Brandon Hayes-Lattin, and I'm going to talk a little bit about the epidemiology and biology of AYA cancers. You already just heard a little bit about AYO Power and our partnership with Elephants in Tea and the goals of this program. So this is the inaugural kickoff, and I'll give some shout outs at the end. But for the talk today, we're going to start with some of the basics of why AYA Oncology really came to highlight as a field, to understand the ways in which the epidemiology of adolescent young adult cancers, both the incidence of cancer and the outcomes associated with the diagnosis of cancer, can differ between adolescents and young adults compared to younger or older cohorts. We're going to use some of these epidemiological trends to understand how we generate hypotheses about how the basic biology of these cancers might be part of explaining the differences that we see. And then I'm going to run through a number of examples where these hypotheses have really borne out, and we've seen unique biology within certain cancer types that differ between younger and older cohorts. And I have no relevant disclosures through this talk. So to begin with, I'm going to go back in time, as Mike said, a couple of decades, to define the AYA population and look at how epidemiology was involved in that. And really, it started quite simple with a series of observations looking at cancer survival rates and how we have or have not improved over time. This is a graph that shows the 5-year relative survival of invasive cancers in the United States and an analysis that was done in 2006. And briefly, what it shows is that for cancer patients who were age 5 to 9 at the time of diagnosis, between 1975 and 1995, every 5-year increment, there was an improvement in 5-year survival rate. Going from a survival rate of approximately 55% up to 80%, and that trend has continued to improve. If you look at the right-hand side, you see that for older patients diagnosed with cancer between age 65 and 69, while the absolute survival rates are lower, there is improvement every 5-year increment, and the graph looks similar, just downshifted. But if you look in the middle of young adults, age 25 to 29 at the time of diagnosis, over that 30-year period, there was no increase in the 5-year relative survival rate. And it was data like this, epidemiological data like this, that began to have people pay attention to what's going on in adolescents and young adults. That led to a very seminal piece of work through the National Cancer Institute and a program they call SEER, which looks at cancer statistics in the United States, and generated this sort of historical graph looking at the rate of change or the rate of improvement in 5-year survival based on the age at diagnosis. And you can see that the improvement rate dropped substantially beginning around age 15 and began to rise again beginning around the age of 40. That led to really a focused attention by the National Cancer Institute, by many healthcare professionals across multiple disciplines, as well as advocacy groups who intuitively know and have experienced the survival challenges of adolescents and young adults, to create a national plan. And as part of that, the field of AYA Oncology was really born. And importantly, it was described as taking care of patients who were diagnosed with cancer between the ages of 15 and 39, again, based on that old epidemiology. I will say that when we look at updates, we do see some trends in the positive and some trends that are worrisome. This takes a look at incidence in the light color of cancer for males and females over time. And what you can see is that from 1975 until the mid 2010s, there has been a progressive increase in the incidence of cancer, both for males and females, across the entire AYA age range. In the more solid colors, you do see that there is a progressive decline in the mortality from cancer. And that is true in the 15 to 19-year-old, 20 to 29-year-old, and 30 to 39-year-olds. Again, the pace of improvement may have lacked compared to younger and older cohorts early on, but it is reassuring that we are making progress. However, I'm going to go through several cancer types today and talk a little bit about how the epidemiological trends have led to questions about why there might be changes in the survival rates or survival gaps, and how basic biology might explain some of those differences. So just quickly, here is, again, from that old SEER monograph, a look at the survival rates for patients diagnosed with leukemia. And the different colors are the age of diagnosis at the time. And what you see is in that initial analysis, there was a major decrease in long-term survival for males diagnosed with leukemia above the age of 15. Really pretty dramatic and striking. Again, kind of leading to that younger age range of AYA as being defined at age 15. But these trends really were seen in multiple cancer types where, compared to young children, once you reach this adolescent and young adult age range, long-term survivors, survival rates were significantly lower and described as a survival gap. So here's a similar curve looking at long-term survival in sarcoma. And again, you see there's a major difference in long-term survival for those diagnosed less than 15 compared to those diagnosed older than that, almost double. Here is a similar graph looking at brain tumors. This is particular to astrocytomas. And again, the cutoff here seems to be around age 15 to 19, but there's a significant gap, almost doubling of survival for young children compared to older adolescents and young adults with this type of brain tumor. But importantly, we see survival gaps at the other end of the age range as well. So while I just showed you examples where there seems to be survival gaps between pediatric patients diagnosed with certain cancers and older patients, this is a look at testicular cancer. Testicular cancer turns out to have a lower survival rate in the 15 to 30 age range and actually improves with increasing age. If you think about most of medicine, that's unusual. Most of the time, older patients don't do as well as younger patients. But here we have an example on the other end of the age spectrum where younger patients are actually having inferior survival. So that was observed in testicular cancer. It's also observed in breast cancer. So this is a look at 5-year survival rates in breast cancer. There's 3 graphs here. One is localized disease, meaning patients who are diagnosed with breast cancer that is localized just to the breast. Regional disease, which means that it has spread into regional lymph nodes, or distant metastatic disease. And again, what you see here is the 5-year survival rates are actually lower for the younger patients. They improve by the time you reach the age 40, the older patients. Again, a little bit counterintuitive and suggestive that there really is something different going on in young patients, young women diagnosed with breast cancer. It's not shown on this graph, but in terms of the epidemiology of breast cancer, it is also true that compared to older patients, young adults are more likely to be diagnosed with regional or metastatic disease at the time that breast cancer is discovered, which is associated with a poor prognosis. So overall, these epidemiological trends led to the definition of AYA Oncology of the study of patients who are diagnosed with cancer between the ages of 15 and 35. And I will say, while that was rooted in the cancer survival and cancer survival improvement rates over time, when this first came out, there was a lot of questions. Hey, isn't that an enormous age range? How can you really lump someone in their teens, in their 20s to someone in their 30s? And I have an answer for that, not only in terms of cancer survival rates, but in terms of other epidemiological trends core to AYA, meaning sex, drugs, and rock and roll. In terms of sex, if you think of the age range 15 to 40, that is really the age of fertility. And that is true across the world, that the age range of female fertility really begins around age 15 and in the 40s drops off. In terms of drugs, here's examples of alcohol and cigarette use. And again, epidemiological trends will show you that beginning around age 15, we see the rapid uptick of the use of cigarettes and alcohol. And actually, by the time you reach middle age, 40s, you begin to see that drop off. And on the left is popularity of music. And unfortunately, you see the epidemiologic trends for rock and roll are that really everyone of every age tends to like rock and roll. But if you do an analysis of hip hop, you see epidemiologically, sex, drugs, and hip hop fit with the AYA age range. So in this first part of the talk, we're really defining this adolescent and young adult age range based on epidemiologic trends, including this classic look at the lack of survival improvements. And from there, the field really goes into a hypothesis generating mode. If we see that there are differences in survival or survival rate improvements over time, we begin to think about why. And I'm going to spend most of the rest of this talk talking about some of the biological hypotheses that might underpin some of the differences that lead to those survival gaps. I'm going to talk a little bit about the types of cancers young adults get and how patients diagnosed with cancer in the AYA age range tend to get a different spectrum of cancers than young children or older adults. Then I'm also going to spend a lot of time talking about the biology of those cancers. When an adolescent or young adult has the same cancer, the same histological cancer as a child or an older adult, maybe some of the differences that the biology of that cancer is different. And then I'm going to give an example of different biology of the patient itself. What is sometimes referred to as the quote unquote host, the patient's biology and how they react either to the cancer or to cancer treatments as potentially being one of the biological hypotheses that explain these gaps. I am not going to talk about a huge number of other hypotheses that might explain these gaps in outcomes for adolescents and young adults. And you're going to hear many more examples of these throughout the series of AYA Power. But just to give an example, you can imagine that there may be differences in outcomes based on differing treatment regimens, different access to care or treatment settings, different components of supportive care, et cetera. So I'm going to move into some of the uniqueness of the cancers that occur in adolescents and young adults. This is a recent epidemiological snapshot of cancer in AYA, age 15 to 39, just in the last decade. And what's really important here is not only what you see in terms of the types of cancer these patients are diagnosed with, but also the types of cancers that you don't see or you don't see very commonly. So what you see here is that there are common cancers in adolescent and young adult like thyroid, brain tumors, testicular germ cell tumors, leukemias and lymphomas, sarcomas, and even melanoma that are classically rare tumors in the population, but actually end up being the common tumors of AYA. You also see that there are some quote unquote common tumors, things like breast cancer and colorectal cancer. And while they do occur, they're relatively smaller components of the AYA cancer burden. So classically, the quote unquote common cancers that occur in adolescents and young adult are different than what we typically see across the population. If you're a male diagnosed with cancer between 15 and 39, statistically, the most common cancer is actually a germ cell tumor of the testicle, testicular cancer. In females, it turns out that across the entire 15 to 39 age range, breast cancer is numerically the most common, but that skews towards the older age of AYA. And we'll talk a little bit about how that differs biologically from older women diagnosed with breast cancer. Digging a little deeper into the types of cancers and the not just the incidence, but the trends over time for AYAs, there's an important observation that the rate of cancer is actually increasing in this population. So among AYAs, the rate of diagnosing cancer has increased almost 30% in the last few decades, raising from 57 to about 74 per 100,000 adolescents and young adults in the US per year. Just back of the envelope calculations, if you look at the census of the 15 to 39-year-olds in the United States, and you see there's about 109 million, that means that each year in the US, there's more than 80,000 new cancer diagnoses in AYA. It's actually approaching 90,000 per year. When we look at these trends, we see that there are certain cancers for which there has been increase, and in bold, significant increase, and that includes among combined males and females, an increase in soft tissue sarcomas, GI cancers, myeloma, and statistically even more significant in kidney cancer, thyroid cancer, and colorectal cancer. At the same time, there's actually been a decrease across both genders in lung cancer. If we dig a little deeper into how the SEER database defines populations as male and female, we see that within males specifically, there is an increase in testicular germ cell tumor, as well as a small increase in astrocytomas, head and neck cancers, and liver cancer, and a decrease in bladder cancer. And looking at trends over time, it looks like this. The sort of top darker blue curve is germ cell tumor or testicular cancer. And if you just focus on that, you see that the incidence per 100,000 has gone from about 5 per 100,000 up to closer to 12. So doubling of the testicular cancer incidence. This is an important curve because you also see this big bump in lighter blue, with the highest incidence of cancer being in the late 80s. That is actually Kaposi's sarcoma, which is directly associated with HIV and uncontrolled HIV/AIDS. You can often find in epidemiological databases that HIV-associated cancers like Kaposi's sarcoma are excluded from the trend analyses because of that phenomenon. It's a little bit harder to see, but right here in gray, there's also an increase and then subsequent decrease in incidence for non-Hodgkin lymphoma. And that's also like largely HIV-related lymphomas. When we look at the SEER database specific to patients defined as females, you see that there has been a statistically significant increase in ALL, in non-Hodgkin lymphoma, and in melanoma, as well as a smaller increase in Ewing sarcoma and pancreatic cancer. But there has been a decrease in extragonadal germ cell tumor, and importantly in cervix and uterus cancer, for which there's effective screening and vaccination. Here's that same trend looking at the incidence per 100,000 in females, and you can see that carcinoma of the breast is significantly the most common. But if you look in the more recent years, you see a significant uptick in thyroid carcinoma as being an increasing incidence in females. Finally, when we talk about what kind of cancers and outcomes occur in young adults through the SEER database, we see important observations based on race and ethnicity. Just to draw your attention to the colors of blue and red, you can see that both for males and females, the incidence of cancer in adolescents and young adults is higher in non-Hispanic whites than it is in non-Hispanic blacks, the blue bar being higher than the red bar. But if you go to the right and look at mortality, it's actually the inverse. The mortality is higher in non-Hispanic Black males and females compared to non-Hispanic white males and females. So again, epidemiological trends showing us differences that need further work. So now that we've taken a look at what kinds of cancers uniquely occur in adolescent and young adulthood, and I showed you that there are some concerns about outcomes, including survival and survival rate improvements over time, let's dig into why that might be in the basis of biology. I'm going to start looking at testicular cancer. Testicular cancer being often described as a prototypic cancer of adolescence and young adulthood, because really the incidence, the onset, occurs around age 15 and drops off substantially around age 40, meaning that there is something about the biology of testicular cancer that leads its incidence to occur smack in the middle of the age range we're describing. It is not entirely clear why that is, although there's been some good work to understand the cell type of origin for testicular cancer. This germ cell tumors arise from primordial cells that that not only occur in the testis, but can occur in other places where these cells were essentially dropped off during fetal development. That includes the retroperitoneum and the mediastinum. So it turns out that across the board, and for adolescents and young adults specifically, patients diagnosed with germ cell tumor, overwhelmingly it presents in the gonads or in the testicle for males. However, as I just mentioned, you can have germ cell tumor of the mediastinum, which this is a picture of. Well, it turns out that with standard cisplatin-based chemotherapy, patients with metastatic testicular cancer have very high response rates and, in fact, very high cure rates. But patients with mediastinal germ cell tumor, while histologically and morphologically it looks exactly the same as testicular cancer, their outcomes with the same chemotherapy regimens are significantly worse. So you might ask, why is that? What's going on with that basic biology? And what's been discovered is that in testicular germ cell tumor, TGCT, as opposed to mediastinal germ cell tumor, there's a really significant difference in a particular gene of interest called TP53, where the rates in seminomas are only 4%, but the rates in mediastinal germ cell tumor are 60 to 80%. In non-seminomas, again, in testicular cancer, only about 20%, but you see 4 or 5 times higher that in mediastinal germ cell tumor. And TP53 is a specific oncogene that is known across multiple cancer types to lead to chemotherapy insensitivity. So again, coming up with hypotheses based on the observations we're seeing that lead to understanding more of the basic biology. So I'm going to run through a number of other cancers with some similar stories. Next, I'm going to talk about melanoma. If we take a look at the epidemiological trends for melanoma, we see something really quite interesting. It is true that the incidence of malignant melanoma increases dramatically with age. And it's also true that, that patients who develop melanoma as older adults much more commonly develop it in sun-exposed areas like the head and neck, which fits with what we think of as the underlying pathophysiology of melanoma, which is multiple recurrent exposures to UV radiation leading to the onset of melanoma. But if you look carefully at this incidence curve in the younger age range, what you see is head and neck melanoma is actually not as common as truncal melanoma. And that flies in the face a bit in the pathophysiological understanding of recurrent UV radiation exposure, and suggests that maybe there's something different in the biology of why younger patients get melanoma, and when they do, why it can often be in the trunk instead of the sun-exposed head and neck. So again, if we look at the basic biology of melanoma tumors and we compare different age ranges, here you see a graph that shows you genetic information for patients diagnosed less than age 30 to 40, 40 to 65, and greater than 65. If I draw your attention just to the top line, you see a gene called BRAF, which is a known proto-oncogene that can occur in melanoma. Well, for patients diagnosed less than age 30, 90% of them have a BRAF mutation, whereas in older patients, it's only 60, 55, 40% BRAF mutation. So nearly double the incidence of BRAF mutation. You see the same thing for the next one down. For P10, which is another proto-oncogene, you see a significant increase, 26% compared to only 6 or 7% in older patients. So in analyzing this sort of data, again, we see a doubling in the frequency of BRAF or P10 gene mutations. I didn't point out, but in that same graph, there are fewer of other mutations, including a gene called NRAS. And when you look at patterns of different genes that are up or down regulated, you actually find that there are a higher proportion of gene mutation signatures that are not related to ultraviolet radiation as the principal cause. And instead, they're related to what's called mismatch repair pathways. So in other words, it's not so much that the genes mutated in young adult melanoma are not associated with UV exposure. It's more that young adults have things broken in the way that they can repair problems in the genes. It's also noted in this kind of whole genome sequencing data in melanoma that some young adult cases, mutations are found not only in the cancer cells, but in what's called germline cells, so all cells of the body, which is suggestive of an underlying predisposition state. Next, I'm going to talk a little bit about breast cancer. And I showed this graph earlier showing that younger women with breast cancer have worse survival outcomes at any stage. So why would that be? Why would young women have worse survival outcomes? Well, in a really classic analysis done many, many years, decades ago, when people looked at the epidemiological trends of breast cancer, they found that there's actually what's called a bimodal distribution. So you see sort of two humps in the incidence of breast cancer. There's an incidence that occurs more around age 40 and then a second incidence that occurs more around age 70. You see that in figure A. In figure B, if you separate patients with estrogen receptor negative versus estrogen receptor positive disease, you can actually see that that bimodal distribution is because there's two overlapping curves, really two different cancers. There's a young woman curve of ER negative breast cancer and an older woman curve of ER positive breast cancer that explains that sort of epidemiological Clemmensen hook or bimodal distribution. It turns out there are other cancers, including Hodgkin lymphoma, that have similar bimodal distributions. So again, here you're taking epidemiological trends of a bimodal distribution and making biological hypotheses around estrogen receptor positivity or negativity to explain the observation. But it goes a bit deeper than that. This is a complicated graph, but if you look at slide B, what you see is the cell of origin is different in young patients. In this case, young is defined as less than 50, compared to older patients in green. You see that what are called luminal progenitors or mature luminal cells are more commonly the cell of origin in older breast cancer, whereas mammary stem cells are more commonly the cell of origin in young women. The graph in A, for those of you who are more engaged in the basic biology, basically looks at cell sorting that shows far fewer of those mammary stem cells in the older patient panels, 61 and 67-year-old, compared to the young patient panels, 27 and 39-year-olds. So this goes to say that perhaps there's a different cell of origin, that breaks and becomes cancer in young adult breast cancer. And it has led to really this pathogenesis model that would suggest that, well, maybe there was a common ancestor of breast tissue, but the transformation event, the event that occurs within the cell that creates cancer, can be different. They can occur in those luminal cells and give rise to a cancer in older or advancing age at diagnosis, including patients who express hormone receptors and or express another protein called HER2. Or the transformation event can happen in these basal cell compartments, a non-luminal cell or stem cell, mammary stem cells within breast tissue. And when that happens, you get a different biology, a different cancer, a basal-like phenomenon, where these tumors occur at younger ages and are more often hormone and HER2 negative. So here we see that in epidemiologic trends. So if instead of looking at that bimodal graph by estrogen receptor positivity or negativity, instead we look at basal-like versus non-basal-like, you see that the luminal or non-basal-like is more common in the older age population, and the basal-like occurs in the younger women who are diagnosed with breast cancer. And in panel D, what you see is that the hazard rate of mortality, the survival outcomes are worse for those with basal-like. So if you have a basal-like tumor at any age, your chances of mortality are higher. But as we see in panel B, those patients tend to skew towards younger women. So again, biological explanations for some of the epidemiological observations we're seeing in breast cancer. If I move on to colorectal cancer, this has been in the news quite a bit lately, that the incidence of colorectal cancer is rising among young adults. And trying to understand why that is, are there basic biological features that might explain it and actually ways to intervene? So one of the things that's been observed is that obesity and what's called dietary insulin load can be different. It's a trend that's changing over time. And there have been, again, some epidemiological suggestions that dietary habits and associated dietary insulin load or obesity are part of the incidence of colorectal cancer. But certainly in this data I'm showing here, if you look at different quintiles, every 20% change in dietary insulin load, there definitely is an effect seen in survival outcomes, whether that be disease-free or overall survival. If we do the same sort of analysis I showed you in these other cancers in colorectal cancer, looking at the genes involved, what you see is in young adult samples compared to older adult samples, there is a skewing of certain genes, including MSH2 and MSH6. These are mismatch repair genes. They're associated actually with a autosomal dominant inheritance and a familial syndrome called the Lynch syndrome. But you can see the rates are 25 to 38% of these mismatch repair mutations in AYA samples compared to only 3 or 6% in older patients. You can see that there's differences in other genes, including EpCAM and PTCH1. In that same analysis, they took a look at biologically plausible mutations, and for each of these genes, you can see the incidence, the percentage, is significantly higher in AYA colorectal samples compared to older adult colorectal samples. So in addition to the sort of autosomal dominant familial syndromes that can lead to colorectal cancer, just the underlying basic biological function of genes within adolescent young adult cancers are different in colorectal. And then I think my last example of the unique biology is in leukemia. But if we start at the most fundamental level and say, who gets leukemia, and when they get leukemia, what type do they get? You can see that the incidence per million of leukemia is actually quite high in infants. then it goes down and starts to rise again around age 25 to 30. And I don't show this, but it exponentially increases with older age. But what's really important in this graph is the different colors represent different subtypes of leukemia. There are chronic leukemias, including CML, chronic myeloid leukemia, which is extremely rare in patients under the age of 15, but you see in kind of the light blue, becomes more and more common approaching age 40, where it's 20+% of leukemia diagnoses. You can also see that ALL in the sort of pale purple blue is the most common leukemia in childhood, but by the time you reach age 20, it is not the most common leukemia anymore. So just at the highest level, you see that age is associated with the onset of different types of leukemia, suggesting, again, different biology of why a leukemia might start based on age. But I'm going to focus just on ALL, acute lymphoblastic leukemia. This is a graph similar to what I showed in the intro that suggests that with each increment of increasing age, the long-term survival is significantly worse. At least it was back in 2006. And again, we start looking at, well, what are the biological features of these ALL cases and how might that differ between adolescents, young adults, and older or younger patients? And might that be part of the explanation? And while it's not really important to understand all the genes here, you can just tell by the colors that there are significant differences in the cytogenetics of ALL in children, where you see in infants this big dark blue, and in young kids you see the orange, which is hyperdiploidy, or translocation 1221 in yellow. Whereas by the time you get into the AYA range, you start to see the dark purple, which is the translocation 922. That's the Philadelphia chromosome, which has really significant both prognostic and therapeutic importance. The other thing that's really important in this graph, though, is if you look at the age we're talking about, 15 to 40, the most common cytogenetic abnormality is actually other, suggesting that these are a group of patients with a different biology for which it's not common cytogenetic abnormalities. It's a smattering of other. So people have begun to really look at that. And what you see here is sort of categories of genetic abnormalities and the incidence of those based on age, childhood, AYA versus older adult. So in yellow, you see childhood, and you see that far and away, you see copy number changes or a particular transcription factor rearrangement called ETV6 RUNX1 as being common. Whereas in the sort of black or gray AYA, the incidence of those is much, much lower. And instead, in that black-gray AYA, you see on the right kinase-driven mutations or transcription factor-driven mutations like PAX5 and not just the Philadelphia chromosome, which is BCR-ABL, but also what's called a Philadelphia chromosome-like genetic sequence being much more common. So this is essentially a way of saying, look, we know there are a variety of genetic abnormalities that can be associated or directly lead to ALL, but the distribution is really very different. And that can certainly be part of the explanation for why outcomes are different in AYA ALL. In this busy slide, I've highlighted in red some of those particular changes. And what you see is things like low hypodiploid or few copies of chromosomes, a particular rearrangement called KMT2A, the BCR-ABL Philadelphia, or the Philly-like mutations. If you look at where it says prevalence for those, you can see they're higher in adults, or at least in AYAs. And if you look at the clinical features, these are the ones associated with poor prognosis. So in ALL, we've really gotten some robust information showing that not only are outcomes different for young adults, particularly compared to children, but there are some very basic biological features that explain why that is. And importantly, the next move in here is to understand not just what happens biologically that is distinctive in AYA cancers, but can we look at what's broken and target it in certain ways? And here you see, again, the AYAs in black have high levels of certain abnormalities for which we may have targeted agents like the drug ruxolitinib that can affect some of these tyrosine kinases. The final example I want to give is not related to certain cancers in the basic biology, but talk a little bit about hypotheses in biology for the patient themselves or the host. And here I'm also going to use an example in ALL. ALL regimens can have significant toxicities, and there was a publication that compared young adults aged 16 to 21 who were treated either in a children's oncology group or an adult cooperative group study of sort of the same backbone therapy for ALL. There are some differences here, but really the point of this is we see a pretty significant incidence, whether you're treated on a childhood or adult trial, of things like liver function test abnormalities, high blood sugars, hyperglycemia, low blood counts, and febrile neutropenia. That's true here in induction. Here is later in therapy after induction. Again, you see liver function test abnormalities, pancreatitis, blood clots, etc. So this can have, these therapies in and of themselves can have significant side effects. But what this shows is there may be underlying reasons within the patient themselves that put them at higher risk of these toxicities. And one example of that is body mass index, where body mass index over 40 associated with obesity is statistically significantly associated with some of these same toxicities. And if you look epidemiologically, we're seeing increasing rates of high body mass index or obesity in young adults. So if you look at, for instance, hepatic toxicity for low BMI, you see only 31% had hepatic toxicity, whereas in the very highest category of BMI over 40, it was double that. It was 62%. That's true for specific lab tests related to the liver as well as hyperglycemia. So again, this suggests that there may be something different about the patient and how they respond to therapy, such as rates of obesity, that can explain some of the outcome differences. So what I hope I've achieved today is to go through these objectives to understand ways in which the epidemiology of young adult cancers differ from young adult and older adult cohorts, to use these trends to generate biological hypotheses that might explain some of these differences, and to discover examples of some of these unique cancer biology. As was mentioned before, this is really the kickoff of the AYA Power. I want to highlight that a number of future presentations are already scheduled, the next one being Dr. Roth on September 13th. So I encourage everyone to stay tuned to this series. And with that, I would like to thank you, and I will pull down my slides and be happy to take any questions.
Brandon, that was a phenomenal talk. I think we didn't realize how daunting a task we gave you, covering all of AYA oncology, epidemiology, and biology. So our apologies, but also just our thanks for kicking off the session. I do see there's a couple questions in the Q&A box. The first question asks about testicular cancer and breast cancer. And do you think that we're seeing it more often because patients are getting screened more often? And I'm sorry, because older patients are seeing doctors more often, getting screened more often. AYAs feel like they're immortal and perhaps not seeing a doctor as early or as frequently as older adults.
So there's a couple of interesting parts, I think, to that question, which is the relationship between screening and cancers in young adults. So while I showed you that the quote unquote common cancers for males is testicular cancer, and common for females is breast cancer in the AYA age range, it's still relatively rare. And so universal screening, things like testicular exams or mammography, are actually not routinely recommended for adolescents and young adults because the incidence of false positives or just no findings is so high. Whereas in breast cancer, the incidence does raise significantly with age. And this has been a moving target, but somewhere between age 40 and age 50, it's recommended that women have routine screening. So what that often means is that for the young adult who does have testicular cancer or breast cancer, that they're not screened for to find early. And even a step further, it may not be on their radar or their provider's radar to even consider or to look for those cancers. And so, yeah, I think that's part of the reason why, as I said, in breast cancer, young adults diagnose more often with advanced stage disease.
And then a follow-up from the same participant is, why are outcomes worse for these patients? Are they just presenting later? You know, you mentioned biology during your talk. What do you think contributes to the poor outcome for these young onset patients?
So the graph that I showed in breast cancer that had survival rates being worse in localized, regional, and metastatic disease, if you look at the number, the survival rate for localized disease is way better than the survival rate for lymph node involved disease, which is way better than patients who are diagnosed with metastatic disease. And so if we see that stage shift at diagnosis, it absolutely explains part of the difference in survival. But in addition to that, stage by stage, cancer by cancer, we see that what's broken biologically in many of these cancers, including breast cancer, is different. And that also explains response rates to therapy and ultimately survival. So I think it's both.
Perfect. There's a separate question asking about subsequent cancers. So most of the talk was focused on initial diagnoses, and there's a question about patients who develop a new cancer as a result of chemotherapy treatment. And is that because the chemotherapy alters sort of the normal cells or the healthy cells and they become cancer cells, or trying to understand underlying mechanisms of developing subsequent cancers?
Yeah. So yeah, first of all, you're right, Dr. Roth. I had a pretty big menu of things to talk about, and I didn't talk about second cancers, but it turns out that really is an important part of the epidemiology of young adults. In fact, if you look at second cancers as its own category, it is also a major category of cancer diagnosis in this age range. The answer is complicated and probably both. There are absolutely well-established patterns of exposure to certain cancer therapies, including certain chemotherapy drugs, as well as radiation, that can then predispose people to develop a second cancer. However, it is also biologically plausible, and there is certain evidence for patients themselves having something unique about their ability to repair damage or otherwise respond to cancer initiating events that make them more prone to cancer. And so if they were prone to cancer to begin with, they're also still prone to a second cancer. So it's probably both.
Perfect. Well, we are getting to the end of the hour, and Scott and I were very clear that we wanted this first talk to be very comprehensive, really open the door up for learning a lot more about AYA oncology. That the subsequent talks are actually going to be cut in half. So they're going to be shorter talks, a little bit more focused on topics that hopefully all of you find relevant. And we importantly want to make sure you know about all of the content that's going to live centrally. So on that note, Scott and I would like to reintroduce Nick, our colleagues from Elephants and Tea, just to talk about next steps, where all of the content's going to live, and hopefully you guys will tell all of your friends, colleagues, and others to really join in on the education session.
Thanks, Mike. And before everyone jumps off, we did just drop in the survey link too, if you could fill that out and give us some feedback. We will be emailing that out to everyone as well. Secondly, Brandon, thank you so much. I know Mike and Scott already mentioned it. You being the first one is wonderful. It was great, but definitely hey, by the way, you're going first. So thank you for that. And Mike and Scott, just thank you again for the partnership with us, with Elephants and Tea and the AYA Education Group. Honored to be working with you all on this endeavor going forward. As we mentioned before, recordings will be available. We'll let people know when those are ready. They will be set up on our website. So we'll send that link out to everyone. That'll be ready to go. Lisa just dropped the link back in there. Brandon already took my job of letting you know what's upcoming, which is great in the series. Our next session is Wednesday, September 13th. Same time, 12 p.m. Eastern, and that is going to be on disparities in AYA Oncology with Mike Roth leading that presentation that day as well. So again, just thank you, Brandon, Mike, and Scott, for all that you do for the cancer community. Thank you all for attending today, for being a part of our first session. It was wonderful. And I want to give a shout out to Kayla Fulgenetti on our team, who's our senior director, having some technical issues. She's been leading the charge on our end. I've been filling her role in this part to end the session today. So just wanted to say thank you for her organizing all of us on the back end to make sure things go off smoothly without a hitch. So thank you all so much. And with that, we can close out today. We look forward to seeing you all in September. Enjoy the rest of your August. And yeah, just thank you so much, and be on the lookout for the follow-up email with recordings and resources. Thanks, everybody.